Imagine you're trying to fit a weirdly shaped puzzle piece into its spot. Molecular docking is kind of like that, except the puzzle pieces are tiny molecules. The goal is to see how one molecule, usually a small chemical compound called a ligand, fits into another molecule, typically a protein that serves as a receptor. Proteins are like molecular machines in our bodies that perform various tasks, and the ligand is like a key that fits into the protein's lock to either start or stop its activity. The process helps scientists predict how drugs will bind to proteins and how effective they might be at fighting diseases. We use molecular docking to speed up drug discovery and development. Testing every possible drug compound in a real lab would take forever and cost a fortune. Instead, scientists use molecular docking to perform virtual experiments on computers. This approach allows researchers to quickly identify promising drug candidates by predicting how well they bind to their target proteins. By doing this, we can narrow down the list of compounds worth testing in real-life experiments, saving time and resources. Let's break down some key terms you'll need to understand before diving into the world of molecular docking. Here's a handy glossary to get you started. Receptor. Think of this as the lock. It's the target molecule, usually a protein, that the ligand binds to. Receptors play crucial roles in biological processes, and their interaction with ligands can trigger important cellular responses. This is the key. A ligand is a small molecule that interacts with the receptor. It can either activate or inhibit the receptor's function, depending on the context. Binding site, the keyhole. This is the specific area on the receptor where the ligand attaches. The binding site is typically a pocket or groove on the protein surface that accommodates the ligand. Conformation. Molecules aren't rigid. They can twist and turn into different shapes. A conformation is a specific 3D arrangement of a molecule's atoms. Ligands and receptors can adopt multiple conformations, and finding the right one is key to successful docking. Scoring function. This is like a judge in a talent show. It's a calculation used to predict how strongly a ligand will bind to a receptor. Scoring functions estimate binding affinity based on various factors such as hydrogen bonding, hydrophobic interactions, and electrostatic forces. Affinity. This term refers to the strength of the interaction between the ligand and the receptor. High affinity means a stronger and more stable interaction, which is usually desirable in drug design. Grid box. This is the virtual space around the binding site where the docking simulation is performed. It defines the area in which the software searches for the best ligand-receptor interaction. Pose. This is a possible orientation and conformation of the ligand within the receptor's binding site. The docking process generates multiple poses, and each one is evaluated to determine the best fit. Exhaustiveness. This parameter controls how thoroughly the docking software searches for poses. Higher exhaustiveness means a more comprehensive search, but requires more computational time. Understanding these terms will give you a solid foundation for exploring molecular docking and its applications in drug discovery. With this vocabulary in hand, you're ready to dive deeper into how molecular docking works and how to perform it using AutoDock Vina. Step 1. Gather your tools. Before you dive into molecular docking with AutoDock Vina, it's essential to gather all the necessary tools. Think of this as assembling your toolkit before building something complex. First, download AutoDock Vina, the main software for performing the docking simulations. It's like the engine that drives the whole process. You can find it on the official AutoDock website. Next, grab MGL Tools, which will help you prepare the input files. This tool allows you to add hydrogen atoms and convert your files into the PDB-QT format, which is the language AutoDock Vina speaks. Visualizing the docking results is crucial, so equip yourself with PyMol or Chimera. These programs are like high-tech microscopes that let you see the 3D structures of your molecules in action. Finally, you'll need the receptor and ligand files. These are the molecular structures you want to dock. You can download receptor files from the Protein Databank (PDB) and ligand files from databases like PubChem or Zinc. Make sure these files are in the PDB-QT format, as this is required for docking with Vina. Step 2. Prepare the receptor. Now that your toolkit is ready, it's time to focus on the receptor, the protein that will interact with your ligand. Start by loading the receptor structure using MGL Tools. Proteins are typically downloaded in PDB format and might be missing some hydrogen atoms, particularly those involved in hydrogen bonding. In MGL Tools, use the Add Hydrogens feature to add these crucial polar hydrogens, ensuring that the protein structure is complete for interaction. Once hydrogens are added, save the protein in the PDB-QT format. This format includes atomic charges and potential rotatable bonds, providing a comprehensive description of the molecular structure. Think of this step as polishing your lock, ensuring it's ready for the key to fit perfectly. Step 3. Prepare the ligand. Next, shift your attention to the ligand, the small molecule that will fit into the receptor. Open the ligand file in MGL Tools and add any missing hydrogens, ensuring the ligand is chemically accurate. This step is crucial because hydrogen atoms can significantly influence molecular interactions. Check the charge on the ligand, as correct charge states can affect binding affinity. AutoDock Tools can automatically calculate the charges for you. If your ligand has parts that can rotate, such as single bonds between atoms, define these as rotatable in MGL Tools. This flexibility allows Vina to explore different conformations and find the best fit in the receptor's binding site. Once the ligand is ready, save it in the PDB-QT format, which includes information about atom types, charges, and rotatable bonds. Step 4. Set up the docking environment. With both the receptor and ligand prepared, it's time to set up the docking environment. This involves defining the search space where the docking simulation will occur. In MGL Tools, you'll create a grid box around the receptor's binding site. This box is essentially a 3D space that guides the docking process. Position the grid box so that it encompasses the entire binding site, allowing enough room for the ligand to explore different orientations and conformations. However, avoid making the box too large, as this can lead to unnecessary computation time and less accurate results. Choose an exhaustiveness level, which determines how thoroughly Vina searches for potential binding modes. A higher exhaustiveness value means a more comprehensive search, but requires more computational resources. Typically, a value of 8 is a good balance between speed and accuracy, but you can adjust this based on your computer's capabilities. Step 5. Now that everything is set up, it's time to let AutoDock Vina do its magic. Create a configuration file that specifies the paths to your receptor and ligand files, as well as the grid box parameters. This file acts as an instruction manual for Vina, telling it what to do and where to look. Once the configuration file is ready, open a command prompt and navigate to the directory containing your files to start the docking process. This command tells Vina to perform the docking simulation and save the results in an output file. During the simulation, Vina will explore different poses of the ligand, scoring each one based on how well it fits into the receptor's binding site. It's like a molecular speed dating event, where each pose is evaluated for compatibility. Step 6. Analyze the results. Once AutoDock Vina has completed its task, you'll receive a range of possible poses for your ligand within the receptor. Each pose comes with a score that gives you an estimate of the binding affinity. The lower the score, the better the fit. In molecular docking, a low score is like getting a gold star. It means the ligand is likely to bind strongly and specifically to the receptor, which is what you're aiming for in drug design. A high binding affinity indicates a good fit, suggesting that the ligand could effectively inhibit or activate the receptor, depending on the desired outcome. However, don't just rely on scores alone. You need to look at the poses themselves. Imagine if your key fits the lock perfectly, but is inserted upside down. Technically, it fits, but it doesn't open the door. Similarly, the ligand should be in a correct orientation that matches known binding modes or makes sense for the target protein. Look for key interactions like hydrogen bonds, hydrophobic contacts, and electrostatic interactions that are crucial for effective binding. On the flip side, bad results are characterized by high scores, indicating weak binding. These are like the keys that jiggle but don't quite turn the lock. Poses that don't make sense might have the ligand outside the binding site or in an orientation that doesn't allow it to form necessary interactions with the receptor. Such poses might be outright rejected or require further investigation to understand why they don't fit. It's important to remember that docking is a predictive technique. It provides a hypothesis about how molecules might interact. While docking can offer great insights, experimental validation is needed to confirm these predictions. This means testing the ligand-receptor interactions in a laboratory setting to see if the predicted binding modes actually occur in real life. Congratulations! You've successfully completed a molecular docking experiment using AutoDock Vina. This process is like solving a complex puzzle, where each piece provides insight into how drugs interact with their targets. Remember, molecular docking is a powerful tool in drug discovery, helping researchers design more effective medicines to improve health and treat diseases. By following these steps, you're contributing to the exciting world of scientific research and innovation.